Hello,
Yes, continue.
am I audible?
Yes. Aishwarya.
Yes, ma'am.
Yes, sir.
Is it visible?
Yes.
Can I start my presentation?
Okay, start.
Good afternoon. I'm Aishwarya Muralidharan, research scholar from Center for Animal Taxonomy and Ecology, Christ College, Irinjalakuda. I'm working under the guidance of Dr. Sudhikumar AV. Here I'm going to discuss about the termite fauna of Asurangundu Protected Area, part of Varani Wildlife Sanctuary in Thrissur district. Termites belong to the class Insecta, order Platodia, and family Termitidae. They are one of the most fascinating eusocial organisms that exhibit cooperative brood care and polymorphism and division of labor. The members of the colony is mainly divided into three castes, namely the reproductive caste, soldier caste, and worker caste. As the name indicates, the main function of the reproductive is to help in reproduction and propagation of the colony, while the soldier caste will function of the soldier caste is to protect the colony from enemies. All other functions to be performed within the colony is done by the workers. Then termites are commonly known as white ants. They are small, soft-bodied insects. Coming to their importance, feeding habits makes them important ecosystem engineers which over long period of time can modify the physical properties of soil such as soil texture, water infiltration, and nutrient content. Uh, Another importance of termite is that their distribution generally varies with factors like temperature, humidity, rainfall, habitat fragmentation, etc., and hence can be used as biological indicators. Disturbances by logging or conversion to plantation or crop fields reduces their relative abundance. So, such sensitivities underline the importance of termite since the soil feeding species with high abundance and biomass suffers the greatest decline when forest habitats are disturbed. So, thus, they can be used as biological indicators. Then, economically, termites pose serious threat to wide range of agricultural crops, timbers, etc., and thus forms major pest to humankind. Coming to the study, the area selected for the study is Asirangundu Forest Area, located in Varani Wildlife Sanctuary, which lies within the Western Ghats region of Kerala. The samples were collected during a period of January to March 2024. Methodology: all the possible microhabitats such as soil, leaf litter, mound, humus on surface, dead wood, trees, all these microhabits were searched for termites, and mainly the soldier and worker caste were collected. And the collection was done using brush or trowels. After collection, dirt will be removed from the sample using a brush. Then it is transferred into alcohol, that is 80% alcohol, and then uh, deposited in storage vials. After this, it will be labeled with the date and place of collection. Then the specimens will be observed under the microscope for identification. Coming to identification of species, the identification is based on morphological characters and is based on the key provided by Chautany and Krishna et al. The main morphological characters used for identification include the shape and size of head, mandibles, antenna, postmentum, pronotum, mesonotum, metanotum, and finally the size and coloration. For the feeding group classification, uh, the guidelines of Collins and Eggleton is followed. According to them, termites belong to five feeding groups, namely the wood feeders, wood litter feeders, soil feeders, soil interface feeders, and epiphyte feeders. Coming to the result, the table shows list of species identified from the area. A total of 12 species were obtained, of which 10 were identified up to species level and 2 up to genus level. From the identified species belong to four subfamilies, namely Macrotermitinae, Nasutitermitinae, Termitinae, and Apicotermitinae. And Macrotermitinae was the largest family to be noted with five species. These are the microscopic images of the specimen collected. The graph shows genus-wise distribution of termites from the study area. Uh, from the area, seven genera of termites were identified, and the genus Odontoterms were the most dominant genera representing 34% of the collection, constituting four species. It was followed by the genus Nasididerms and Microceroterms representing 17%, each with two species. While noticing the feeding groups, there were two main feeding group of termites. First one is type 2 feeders, that is the wood or litter feeders, represented by the genera of Odontoterms, Microterms, Microceroterms, and Nasutiterms. And another type of feeding group found was type 3, that is the soil humus feeders, and was represented by the genera of Euhameterms and Speculatums. So, studies have shown that soil feeding groups cannot survive in a disturbed ecosystem, but wood or litter feeders can better survive in disturbed areas that provide high organic content. So, from this present study, soil feeders have been reported, indicating that the area is less disturbed. And this preliminary study can be used as baseline data for future surveys. These are the references and acknowledgement. Thank you. Yes, sir.
I have one nice presentation. I have one question. Uh, Good feeders you mentioned. Good feeder termite. Good feeder.
Yes, sir.
Okay. So in that good feeder termite, which family belongs to that good feeder? Micro. Okay. So is there any preference for certain plant species? I mean, certain bark preference is there for that particular family, for that particular termite family? Is there any preference for bark? I mean, bark preference is there? Means the good of
Hello,
this.
sir. Audio is not clear.
Uh, Actually, is there any preference for book in the sense this particular term you prefer, for example, manticoras for terminators, that kind of preference is there, whether you observe it or not.
Group 2 feeders are usually soil feeders.
Aishwarya,
Yes,
Aishwarya, can you hear me?
audio is not clear.
Aishwarya,
Yes, ma'am.
how was the authentication of the species done?
Identification was based on morphological characters.
Authentication. Identify using morphological characters, but authentication might be showing some references.
It was based on key provided by Chortany in 1997 and Krishna et al. In 2013.
Is there any future prospect for your study? You have to mention.
Ma'am, can you please repeat?
Is there any future prospect you have to mention? Future study.
Ma'am, it's not clear.
What would you like to do in the future? How would you like to carry on your work in the future? So, Keshwarya, you have presented very well. Thank you. All the best.
Thank you, ma'am.
Next, Reshma Baby. Ecology guys, college irrigation. My topic is I would like to present a review on the current status of development of Waha in India. We all know that centipedes are one among the oldest terrestrial predators of Earth.
Their lineage can be traced back to around 400 million years, but they are very overlooked species. Yet they are very overlooked species. So today, I would like to throw light on these very fascinating species. So centipedes, comprising of the class Chiropoda from the subphylum Myriapoda, are those ventrally flattened, bilaterally symmetrical, mesometrically segmented animals. They are known from all continents except Antarctica with their greatest diversity in the tropics. Um, we all know that centipedes are generally known as a menace, but because of their painful bite and their web. Um, but recently it is found that biological studies have proved that their venom has anti-tumor or antimicrobial as well as analgesic properties. So this has proved that they are a significant group in medical and toxicological research. Sediments constitute a pivotal fraction of the biomass of the macroinvertebrates of the soil fauna. They are primarily nocturnal and prefer the deep hydrophilus microhabitats. They exhibit accelerated growth during the rainy season, and many of the species are found to adhere to annual life cycle. They are active predators that regulate the population of various invertebrates, and are also and they also act as prey, so that they are unable to grow in the food web. So the taxonomic study of Scolopendropopa in Kerala began with a description of Hormosatalus by Werhoff in 1973. That was from Nirmal Garden. And in 1984, Jackie and Gus described digital species from in a paper titled Scolopendropopa of the. Then the first comprehensive effort to catalog Kerala's Scolopendropopa was made by Dr. P. M. Sureshan. That was back in 2003. In 2006, he provided an illustrated key and also updated the checklist of Kerala's Scolopendropopa. Dhanya, Dr. Dhanya Balan and Dr. V. S. Sureshan was the first to identify Laminotubidae from Kerala, and they identified a new species, Cryptops malvarensis, and that was named because it was identified from Malabar wildlife sanctuary. Recently, in 2003, Rahul Krishnan and Dr. G. Prasad, they conducted a study on the reserve forests of southern Kerala, and they documented around 14 species across 3 orders. The current status of Chiropoda, there are more than 3,000 species of Chiropoda described across the world, and more than 700 species of Scolopendropopa described globally. In India, the class Chiropoda comprises of 77 species distributed across 3 orders. Um, and among these 37 species, 75 are from the order Scolopendropopa. Dr. Vinod Khanna in 2008 checklisted around 90 species of Scolopendropopa from India, but due to several changes of your AZ taxonomy and other changes, the number of listed species has been reduced to less than 80. The state of Kerala harbors ground vertebrate species distributed across 2 families, Scolopendridae and Cryptopidae. There are 5 globally recognized families in this order, but only 2 families are identified in India and also in Kerala. The families Scolopendridae generally exhibit a taxonomic representation with around 7 genera, whereas family Cryptopidae is less diverse and are represented by 2 genera. Some species like Ergospinus calciumalensis or Rhizina peltana or Photocyclus dabili are found to be endemic to the state of Kerala. The most recent addition to this group is that of Rhizina peltana made by John Joshi in 2019. That was from the Shepherdly Wildlife Sanctuary, Kollam. Kerala, located in the Western Ghats, harbors a diverse ecosystem that support a rich diversity of sediments, but they are largely overlooked. So it is high time that we should have a comprehensive field research and accurate species identification so that we can have a better knowledge on their distribution, their ecology, endemism, and also we can develop better conservation and management strategies. Thank you.
Reshma, nice presentation. Okay. It's a review one. Okay. So you mentioned that endemism about your conclusions line. So what are the endemic species that you observed during the review? Endemic species. How many?
So Lycica purpura, the introduction is an endemic species that is not exclusively in Kerala. Then,
Like Kerala or Sri Lanka?
so in Kerala.
Kerala.
So certain other civilizations in Western Ghats, then nitrificators in to the South Indian part, and also gravity or those things they are in Kerala.
So, so out of 77, three or four endemic.
So there are,
Then why you opted a topic? Oh, what specialty that you found?
as I mentioned, they are highly evolved species. They in Kerala, as a part of Western Ghats, we have high diversity, but they are highly evolved. And as a part of my PhD was I'm doing the space of scientific.
Okay, the diversity of individuals. Okay. Okay.
Ishma, you have done case study, extensive review of the debates of Kerala. I appreciate for that. How many publications would you have referred, or what is the timeline of publications? The first and last one.
The Adidas publication was from 1984. That names as a key even now. From that, Jardi Joshi's publication in 2019 or Raghu Krishna's publication 2023.
And it is the recent one. So, is there any cultural threats scientifics are facing nowadays? According to you, what might be the threats?
The major threat is they are not yet, they are not observed or they are not yet considered as a important species. Okay? They are considered as endemic, like they have venomous bites and they are not given proper consideration.
On this question.
Thank you, Reshma. Next, we have Vidya.
Okay, good afternoon. I'm Vidya. I'm a faculty from Census College in Malappuram. So, I'll present on the topic unraveling the mechanism of fragility of PCL6 breakpoint region in diffuse large B-cell lymphoma. Actually, this is a part of work which was published recently, and I thought of sharing a small part of this work. Getting into the topic, diffuse large B-cell lymphoma. So before getting into the topic, I just want to let you know a brief introduction about the DNA double strand breaks and the chromosomal translocation. So DNA double strand break, you would have heard about the DNA breaks, both pathological DNA breaks and physiological DNA breaks. Pathology is the one which is harmful to the body, which is easily repaired by we all have a efficient DNA repair system and easily repairs the DNA there. And there are physiological variables which happen, which is needed for some part of the body process. So here, chromosomal translocation happens only when the DNA breaks if it is not repaired properly. So if they are not repaired properly, the genes after breaking, they can get swapped. If two chromosomes break at the same time, the genes can swap between each other. And if the genes are coming under a wrong promoter, they can get activated and in some cancer cases, they get overly expressed. So this is what happened in the case of diffuse large B-cell lymphoma. This is a lymphoma of B cells. You have heard about B cells. B cells are major responsible cells for antibody selection. So the uncontrolled proliferation of B cells is the responsible factor for diffuse large B-cell lymphoma. There are two reasons why this cancer thing are reported, two major reasons. One is chromosomal translocation, other is somatic hypermutation. So, with somatic hypermutation, you would have heard about B-cell recombination that is happening in the immunological cells, which is needed for antibody production. So, there physiological DNA break happen. So, there introducing variability in the antibodies, somatic hypermutation physiologically happens in them. So, that sometimes when this somatic hypermutation abnormally happens in these cells, it will result in the problem activation of certain autogenes. Another is BCL6 chromosomal translocation. So my focus here is the particular gene that is BCL6. Why this gene is important is it's a master regulator of B cells. That is, when physiological DNA breaks happen in the B cells, this particular gene will suppress certain apoptotic proteins. Apoptosis, the cells which, if it is not repaired properly, the cell has to undergo cell death. And if it is not happening, and this particular gene will suppress those genes because to prevent the false alarm. So what is happening in this particular cancer, this particular gene is overly expressed. And how it is overly expressed? It is getting translated to different chromosomes. And in all the cancer patients, BCL6, most of the patients I would say, BCL6 is the common gene which is very important compared to other genes. That's why we focus on this particular gene, that is BCL6. So when we analyzed the patient samples, we found that the vital UTR region of this particular gene, most of the breakpoints, patient breakpoints falls in the spiral UTR region. So what is the peculiarity of the spiral UTR region of this particular gene? So we analyzed it, we did a literature survey, we studied what would be the reasons of gene fragility, and we found some of the studies which is giving implications on non-DNA structures. We all know that normally DNA exists as DNA forms, the stable DNA conformation. But sometimes DNA can adopt a non-DNA conformation. But usually when they adopt a non-DNA conformation, there are certain disorders will restore the structure. But sometimes in these cases, these structures are so stably formed in the cell that they can be easily attacked by nucleases. And what we found is the particular part of nuclear region of BCS gene has a number of non-DNA structure formation, which is proved by different experiments. We took out that particular part of the gene, and this is the science of oligo rat studying in vitro experiments. And there we found that this particular section or part of the gene has the ability to form stable non-polymer structures. And some cases, these structures are not resolved properly by resorbases in the nucleus, and that's when this happens. The BCS will break, get easily translocated to, the BCS, the particular part of the BCS will break and it easily translocated to different other genes because, in the B cells, physiological DNA break happens, recombination happens, so for that chromosome protein will break for sure. And at the same time, if BCS gene also break, and if it is not repaired properly, they can get swapped. And BCS will come under the control of the gene which is present in the chromosome protein, which is normally already expressed in the B cells. And BCS will already express because after some time BCS has to be suppressed, and it is not suppressed only after a particular time, it will be already expressed, and that will cause this particular cancer that is BCS, diffuse large B-cell lymphoma. So as a whole, what our study put forward is that there is a chance of stable non-DNA structure formation inside the cell, and when multiple factors like DNA repair also has not happened properly at the same time, it can result in chromosomal translocation and overexpression of genes. Not all genes, if the genes are oncogenes, then only it will be a problem. And that is the result of diffuse large B-cell lymphoma. So present study shows the region of BCS is cluster because we divided that particular region into different clusters. and can form non-binary structures like heparin, G12 plus, etc. Research speculate that such non-binary structure when present within the genome can make the region or the genome fragile. So thank you. I hope I could make the point.
Your thesis on title.
My title was the mechanism, understand the mechanism of chromosomal translocation in diffuse large B-cell lymphoma.
In that slide, you show the translocation of BCL6. That's from your study or?
Yes, my study.
Okay, okay. Oh, nice man. Excellent. And one more thing, the patient sample, you took patient sample in a
this patient sample, this analysis was done the PCR analysis of patients was done previously and is reported in the literature. So we took the analysis, everything, and we checked where exactly different patients the breakpoint has fallen. There is a PCR of the patient samples, but it's particularly PCR. And the sample sequence they have put in the database. So we took from the database, we took the genome sequence of the patients and we analyzed each and every paper exactly in all the cases, patients where exactly the rate has seen in the PCOS gene. And when we analyzed in almost all the patients, the break is always at the same point. That is the 500 area region of the PCOS gene.
For that also, whether you have any collaboration with the
so actually, I have worked with in IISc in the Institute of Science. So the facilities, the individual facilities which I could do is the same.
Whether it's in CCND, no.
No, I complete work in and for bioinformatics analysis in the bioinformatics institute. I lead the bioinformatics.
Nice man.
Yeah.
Really nice presentation with them, and they even answered all the questions which I have noted. Whether the study was in virtual or in my
both in vivo also, in vivo in the sense we recombined this particular BCS gene in a plasmid, we reduced inside the cell and then we did some of the inversions. I will not explain those experiments in detail.
And I also had written work, which is the methodology used for gene expression studies and the topic was for bioinformatic analysis.
Bioinformatic analysis, it was, just a second. Input analysis, so input analysis usually.
Thank you.
Thank you. Thank you.
Next, Jyoti.